This lecture covers development of the somites and neural tube. Formation of the nervous system begins during the third week of development with differentiation of the neural plate and subsequent elevation of the neural folds. Fusion of the neural folds into a neural tube begins in the neck region of the embryo at about the level of the 5th or 6th somite and extends both cranially and caudally. Until fusion is complete, the neural tube remains open and in communication with the amniotic cavity at both the cranial and caudal ends. This area is referred to as the cranial or anterior neuropore and the posterior or caudal neuropore. Closure of the cranial neuropore occurs at approximately day 25. Closure of the caudal neuropore occurs at day 27. Once the neuropores have closed, neurulation is complete, and the future central nervous system, i.e. the brain and the spinal cord, is represented as a closed hollow tube. Occasionally, the anterior or posterior neuropore fails to close and fuse. When this occurs, the anterior-most portion of the brain, or the posterior-most portion of the spinal cord, remains splayed open on the dorsum of the embryo and eventually degenerates due to exposure to amniotic fluid. Failure of the cranial neuropore to fuse results in anencephaly, which is the absence of a major portion of the brain. Failure of the posterior neuropore to fuse results in rachesthesis. During neural tube formation, cells of the neural plate rapidly divide to form a thickened neuroepithelium. The neuroepithelium gives rise to all of the neurons housed within the central nervous system, meaning the brain and the spinal cord, including the cells that support them, meaning the glia cells. Spinal nerve development occurs in association with development of the somites. The somites are segmented blocks of paraaxial mesoderm that extend in series from the future occipital region of the head to the caudal end of the embryo. Normally, 42 to 44 pairs of somites are formed. The first 4 somites will contribute to structures in the head, whereas the caudal-most 5 to 7 will degenerate as the embryo loses its long tail. The remaining 31 to 33 somites will contribute to the formation of the axial skeleton, skeletal muscles of the body wall and limbs, and the dermis of the skin. The epidermis, however, is derived from ectoderm. By the beginning of the 4th week, 2 distinct populations of somitic cells are recognizable. Sclerotim cells dissociate from the somite and migrate medially to surround the notochord and neural tube. These cells will give rise to the bones and connective tissues of the vertebral column. The remaining cells of the somite form the dermomyotome. These cells give rise to dermis via the dermatome component and skeletal muscle via the myotome component. Once formed, each dermomyotome will subsequently divide into a dorsal or epaxial block and a larger ventral or hypaxial block. The dorsal or epaxial cells will migrate to a position beneath the dorsal ectoderm and give rise to the dermis and intrinsic skeletal musculature of the back. The ventral or hypaxial cells will migrate ventrally into the somatic layer of the lateral plate mesoderm to give rise to the dermis and skeletal musculature of the ventrolateral body wall and limbs. The neural tube segment or spinal cord segment that's opposite each somite is devoted to sending motor impulses to all of the skeletal muscle fibers derived from the myotomal portion of that somite. Likewise, it also receives sensory information from those same muscle fibers. And it is also responsible for receiving sensory information from the sensory receptors located in the patch of skin derived from its dermatomal component. Formation of the vertebral column begins during the 4th week of development, when the sclerotomal cells on each side migrate around the notochord and developing spinal cord to merge with cells from the opposite side. Once in place, each cluster of sclerotomal cells splits to form a cranial and a caudal portion. This splitting allows for passage of the developing spinal cord and its rami. The caudal half of each sclerotome subsequently fuses with the cranial half of the sclerotome below to form the primordium of a single vertebra. As a result of this process of resegmentation, each vertebra is formed from 2 adjacent somites. Now that we've discussed how the vertebrae are formed, let's move on to the intervertebral discs. So the centrum or body of each vertebra is formed by the sclerotomal cells that surround the notochord. As these cells begin to differentiate, the notochordal cells in the region of the body of the vertebra regress. And eventually disappear. The notochord, however, persists and enlarges in the spaces between adjacent vertebral bodies. Here, the notochord will form the nucleus pulposus, or inner core of the intervertebral discs. The tough circular exterior of the intervertebral disc, or annulus fibrosus, is derived from the sclerotome, as are the associated vertebral ligaments. The vertebral arches of the vertebra are derived from the fusion of the right and left sclerotomal populations. This fusion event, as well as the subsequent differentiation into bone, is dependent upon signal received from the underlying roof plate of the neural tube. Failure to receive these signals results in the failure of the bony arch to form, a condition known as spina bifida. Several different forms of spina bifida are recognized. Drawing A illustrates spina bifida occulta, which is the most common form of the condition and is present in up to 24% of people. The defect is restricted to the non-union of right and left lamina from 1 or 2 vertebrae. The skin adjacent to the bony defect continues to develop properly. In some cases, the location of the bony defect is indicated by a thick tuft of long hair. Individuals with spina bifida occulta are usually asymptomatic. Images B and C depict spina bifida cystica, which is characterized by the non-development of 1 or more vertebral arches. This leads to herniation of the vertebral canal contents through a defect in the skin at that location. 2 forms of spina bifida cystica are recognized. Drawing B illustrates spina bifida cystica with meningeocele, which is when the herniation involves only the spinal meninges, which are the membranes that line the vertebral canal and encase the spinal cord. Drawing C depicts spina bifida cystica with meningeomyelocele. This type of herniation involves both the meninges and the spinal cord. Neurological symptoms are usually present in severe forms of meningomyelocele. Drawing D depicts rachis, which is a neural tube defect but not a form of spina bifida. This condition occurs when a portion of the neural tube itself fails to fuse, leading to improper formation of the spinal cord. These two pictures illustrate the two forms of spina bifida. Notice the tuft of hair present in the patient with spina bifida occulta in comparison to the much more serious herniated vertebral contents in spina bifida cystica. This concludes our lecture on the development of the somites and neural tube, as well as the entirety of the embryonic lecture series.